Hi, everybody. I'm Sarah Crump. I'm a new assistant professor in the department, and I'm going to share just one research snapshot, a little vignette, that is a little project I've got with my former postdoc advisor, Beth Shapiro, and geochronologist Terry Blackburn, both at UC Santa Cruz. This project actually started as a Little League sideline conversation between Beth and Terry, and really started with the simple question, can we get ancient microbial DNA out of mineral precipitates that formed under ice sheets? So Terry has this big collection of subglacial precipitates, most of which are from the East Antarctic ice sheet, some from the Barnes ice cap, which is the final remnant of the Laurentide ice sheet. These mineral precipitates form under the ice due to changes in subglacial hydrology and due to the concentration of solutes as liquid water freezes to the base of the ice. And this formation is thought to be really rapid, and it really locks in this incredible snapshot of these subglacial waters. So Terry has dated these samples with uranium lead and done lots of great work on them. You can see we're working with samples that are late Pleistocene in age, up to about 500,000 years old. So Terry handed off about 30 of these dated samples to me with the task of trying to get microbial DNA out of them to learn about the microbes that have lived in these subglacial waters. And these are incredibly hard to access environments. So it'll be very cool if we can do this. So because we're working with trace amounts of degraded DNA, we're following these strict ancient DNA protocols to maximize our yields and minimize contamination. Here's a quick plot of those DNA yields. We've got concentration on the y-axis and age on the x-axis. The colors denote the quality of the DNA libraries that we're making. So you can see the younger samples behave better. We're getting more yields out of these young samples and then a drop off with age. This makes good sense and actually gives us some confidence that we're getting some real endogenous DNA out of these samples. But to really know what's going on, let's take a closer look at the taxonomic results when we actually sequence these samples. So this is just one sample. The taxonomic assignments in this plot, higher taxonomic ranks are in the center of the circle. So you can see this sample and all the others is dominated by bacteria, with about 10% assigned to archaea. There's definitely not enough time to go through the full results, but I've pulled out listed a few taxa of interest here. These are representative, relatively abundant taxa that we see in these samples. And what's important to note is that these are all cryophiles. These are taxa known to thrive in cold aquatic environments. So this makes great sense for the settings in which these samples formed and gives us more confidence that we're actually getting a genuine signal of the subglacial microbiome and learning about who can eke out a living in these crazy subglacial worlds. So thank you, more to come, but thanks for listening.